Today is the first class of theory of dermatology, skin and aesthetic. Before starting, I should say something. We will discuss the basic about all the dermatology and the basic skin lesions in our practical classes, and we will show you the cases also. But for the theory class mainly, except today's class, we will focus on the last 10 years questions that have arisen in West Bengal Health University. So those questions are really tough, and for that we will give you just point-wise notes that you will need to write, and practical will be separate. Now today's class will be about the basic anatomy of the skin, and although I have taught that to all the batches except the last batch that didn't come due to the corona, and for their benefit we will take the class. Now coming to the basic anatomy of the skin and skin, we have the three layers of the skin, that is epidermis, dermis and the subcutaneous fat. Most important for us will be the epidermis, and we'll discuss in detail about that. We will have 5 layers in the epidermis: stratum basal, stratum lucidum, which is only found in the palms and soles, stratum spinosum, stratum granulosum and stratum corneum. The topmost layer will be stratum corneum, then will come stratum lucidum, then granulosum, spinosum and basal. Dermis is now separated into 2 parts, the papillary dermis and the reticular dermis, and then we have the subcutaneous fat. As you can see, these are the layers of the skin. The upper part till this part is the epidermis, and these are the gritty ridges that is coming down, and the part around the gritty ridges, the dermis is called as the papillary dermis, and the lower part is called the reticular dermis. The lower part, 2/3 of the dermis is the reticular dermis, and 1/3 is the papillary dermis, and then we have the hypodermis or the subcutaneous fat. This is another picture with the appendages. These are the hairs with the hair follicles and the sweat glands, and we have the sebaceous gland also. This is the sebaceous gland, and this is the muscle that attaches through the hair, and it's got a pyroerective muscle, and these are the blood supplies. We'll tell you the blood supplies in detail later. Now coming to the layers of the epidermis. As we can see, the first layer is the stratum corneum from top. This is a full dead layer. It is made up of dead keratinocytes, which are more flattened and horny layer of keratinocytes. Then we have the 2 or 3 cell thickness stratum lucidum, found only in the palms and soles. Then we have the stratum granulosum, which is named because there are a lot of granules, keratinohyaline granules. And then we have the stratum spinosum. Stratum spinosum is due to the lot of spine-like projections of the cells because of the attachment of one cell to the other cells through desposomes. And the most important layer and the only living layer of the epidermis is called the stratum basal. Now stratum basal is the only germinative layer, and it has certain other cells in it also, like melanocytes and the Merkel cells in it. What are the types of cells in the epidermis? We have the keratinocytes, we have the melanocytes, the Langerhans cells and the Merkel cells. Now stratum basal, it is made up of cuboidal columnar cells which has large oval nuclei and dense basophilic cytoplasm. Stratum spinosum is also called a spinous or the prickle cell layer. It is formed with polygonal cells with delicate processes. Desposomes connect the adjacent keratinocytes, and this gives the rise of a spinous-like process. Now what happens actually is that most of the cells are circular in shape, but once it is attached to other cells, it forms like spinous-like processes, which is not seen usually, but if we put it in a hypotonic solution, we can properly see the spinous layers better when the cells shrink. And these are the polygonal cells with delicate processes. Desposomes connect the adjacent keratinocytes. Then we have the stratum granulosum, which has flattened diamond-shaped cells filled with coarse basophilic keratinohyaline granules. Now we have the stratum lucidum, which is the clear layer found in the palms and soles, and which is 2 or 3 cell layer thick. We have the stratum corneum, which is a flat anuclear eosinophilic corneocytes, dead layer shed during epidermal turnover. Now, what is the epidermal turnover or the transit time? It is the time taken for a cell to pass from the basal layer of to surface of the skin. Now, what happens usually is that in the basal layer, we have the daughter cells, daughter keratinocytes, and these cells, they divide and form the cells up, like the stratum corneum, lucid spinosum, and the gynosum, and the stratum corneum. As it goes up, there are certain changes of the cells. The cell organelles dies off slowly, and the cells become more flattened and has less cell organelles. And this full change is called as keratogenesis. This is a morphological change. And the time taken for one cell to go from the basal layer to the topmost layer is around 52 to 75 days, out of which the mitosis that happens in the basal layer takes around 16 to 24 days. Now, what are the other cells that are found there? The most important is the melanocyte. All our skin color, the racism that we see nowadays, is due to these melanocytes. This imparts the color of the skin. Now, people who are more toward the exposed to more sunlight, or who are present more near the equator, will have little bit more number of melanocytes. Although the numbers doesn't really vary, the cell organelles that are melanosomes, which has the molecule which imparts the color of the cells, these are bigger. Not only bigger, they are more in number in those people. In fairer skins, the melanosomes are much smaller and less in number. So what are melanocytes? They are neural, and remember, each melanocyte in one basal layer will supply to 36 keratinocytes. Now, what are melanocytes? With their neural crest-derived cells, the dendritic cells that synthesize and secrete melanin contain organelles called as melanosomes. And they are located in the basal layers 1:10 ratio. That is, for each 10 cells, daughter keratinocytes, we have one melanocyte in the basement membrane. And then with the epidermal melanin unit, a single melanocyte supplies melanosomes to 36 keratinocytes. This is important for you for the fiber. And melanocytosomes vary in number and size according to the skin type that I already explained. So this is a picture of the melanosome, and you can see all these are melanocytes. Sorry, this is the melanocyte and these are the melanosomes, and this you can see it is supplying to the surrounding keratinocytes. And it is found only in the stratum basal. Now, melanin are formed through mediation of tyrosinase and dopa. What are the functions of melanin? They impart color to skin and hair. They protect the skin from UV radiation, and it is a biochemical utilizer of toxin, free radical, oxygen derivatives. Now, what are the other cells that are found? The others mainly cells with the Langerhans cells, with the APC, the antigen producing cells. These cells are the first responders for any infection that happen in the skin. These are type of macrophages. The role in various immune processes like allergic conjunctivitis and immune tolerance and surveillance against neoplasia are all done by the Langerhans cells. Now we have the flat neural cells which are called as Merkel cells, and these are neuritic cells which mainly respond to touch, and it detects mechanical deformities of the epidermis, and so it also helps in the immune process also. So here you can see the Langerhans cells, which also has dendritic-like processes, and we have the Merkel, which is not given in the picture. Now, coming to the physiology, what are the functions of the epidermis? It is mainly a cornification. That is, it will give a hard appearance of the skin, and it has barrier function. Obviously, it will cause a barrier from the outside germs to enter in the body. The permeability is very important. Mainly the transepidermal water loss. The function of the epidermis, it will control your body temperature. It will allow the amount of water that will be evaporated out of the skin, and hence it will control the body temperature, which is very essential for all the functions of the body. Maintenance of fluid and electrolyte balance. We have thermoregulation, the pigmentation of the skin, the immune function, sensory receptors, and obviously the vitamin D synthesis. Now, as vitamin D is now considered as a hormone, so skin is basically one of the biggest hormonal organs, endocrine organs of the whole body. It is the biggest. Skin itself is an organ, remember that. Now coming to the dermis, we have the papillodermis and the reticular dermis. The papillodermis is thin zone beneath the epidermis and is only 1/3 of the total dermis. The reticular dermis, now see the picture. We have in the basal layer, we have these big VT ridges. You consider the picture. And above this is the epidermis. So below it is the dermis. So now this part of the epidermis is the papillary dermis. And below this, the 2/3 part is the reticular dermis. And below this is the subcutaneous fat. The reticular dermis, all the blood vessels, the interconnecting blood vessels, the arteries and veins are present in the epidermis. There are 2 layers of that interconnecting arteries and veins, and they give rise to arterioles, which goes up, capillaries, which goes up to the papillodermis and supply there, and then come down as other capillaries. Now, none of the blood vessels or the capillaries goes to the epidermis, because basically the epidermis is dead, so there is no need for blood vessels there. All the nutrients that is there in the epidermis come from the dermis, so the diffusion from the capillaries. So it is the only basement layer that gets the blood supply. So as you can see, this is the papillodermis, and this whole thing is the reticular dermis. We have the stratum corneum, stratum lucidum, stratum granulosum, and stratum spinosum, and stratum basal. And these 2 layers, stratum spinosum and the stratum basal, together is called as stratum germinosum. And in the hypodermis of the subcutaneous fat, we have the adipose tissue and the areolar tissue. What is the structure of the dermis? It is a non-cellular connective tissue. It is constituted of collagen, elastic fibers, and ground substance, mainly mucopolysaccharides, chondroitin sulfate, and glycoproteins. It has embedded nerves, blood vessels, lymph vessels, muscles, and pilosebaceous unit, the apocrine and the eccrine units. The cellular content includes fibroblasts, mast cells, histiocytes, Langerhans cells, lymphocytes, and eosinophils. Now these Langerhans cells are present both in the epidermis and the dermis. Now, what is the variation of the thickness of the skin? The difference, there will obviously you can see there are a lot of difference between the thickness of the skin up in your body. The thickest are in the palms and the soles, that is around 1.5 millimeter, and the thinnest is in the eyelids and post-auricular area, which is around 0.5 millimeter thick. Obviously, children and elderly will have thinner skin, and basically for males will have thicker skin than the females. This is one of the most important structures that we will read in the anatomy, that is called the dermoepidermal junction. All the deposition, all the supplies, blood supply, and the transfer of nutrients occur in this. We have a lot of diseases that deal with the dermoepidermal junction because the antibodies found in the DEG, mainly like bullous pemphigoid. that will come and teach you in the later classes. Now what does DEG contains? It contains the basal lamina, the lamina lucida, the lamina densa, the anchoring filaments, the anchoring fibrils and dermal microfibril bundles. What is the main function of DEG? The main function of DEG is that it holds the epidermis with the dermis, and also it will help all the blood supply and the nutrients to go from the dermis to the epidermis. So this is a big picture, this picture of a DEG. Now this is the DEG. These are the desmosomes. And we have the dermal collagen, type 1 collagen, the anchoring proteins, the anchoring fibrils. We have the lamina densa, the lamina lucida, and the fibroreticular lamella. These are the lamina. It is a very complicated picture, but you should know how to draw it in case the question comes. Although in theory we don't have questions. I didn't see any questions in the last 10 years from the anatomy of the skin. But if you don't know the anatomy of the skin, you will not be able to explain in the diseases. And for some diseases, when there is a 5 marks question for you, you have to first describe the anatomy of the skin, then only you will be able to describe the disease. What are the functions of the DEG? There is the attachment of the dermis to the epidermis. They are most important. Support to the epidermis. Regulation of permeability. Autoantibodies to protein in the DEG may be responsible to vesicular disorders like bullous pemphigoid, which is due to autoantibodies due to BP 120 and 180. There are two different proteins. What is the embryology of the skin? All constituents derive from ectoderm and mesoderm. Ectoderm and mesoderm begins to proliferate and differs in the 4th week of interuterine life. The specialized structure, the most specialized structure of the skin are the teeth, hair, nails, and the glands beginning to appear at this time. Now coming to the appendages of the skin. We have certain appendages of the skin like hair follicles, the sebaceous gland. Now hair follicles with the sebaceous gland together is called as the pilosebaceous unit. We have the apocrine glands and the eccrine glands, which are the sweat glands. Now hair. What are the types of hair? We have the lanugo hair that is mainly found in the interuterine life. Sometimes the babies are born with lanugo hair, which are shed off. We have the vellus hair, which is a soft, thin hair found in the body. And then we have the terminal hair, like the scalp hair and the hair in the groins. What are the size of the hair follicles? It is found over the entire surface of the body, except the palms, soles, the glans penis, clitoris, labia majora, mucocutaneous junctions, and distal portions of the fingers and the toes. Now coming to the end of the hair, just you need to know in case they ask you. We have in the longer section it is the infundibulum, the isthmus, the stem of the hair, and the bulb. And in the cross section we have the outer sheath, inner sheath which consists of the hairless layer, Huxley's layer, and the cuticle. We have the cortex and the medulla too. Now phase of the hair growth. Why you need to know this? Because there are different diseases to different hair cycles. What we see actually the hair is the anagen phase, which is the growing phase which lasts for 2 to 10 years. About 90% of the hair are in the anagen phase at any given time. We have the catagen phase, that is the inversion phase, lasts for 1 to 3 weeks, and about 1% of the hair are in catagen. And hence, and the telogen hair is the resting phase, lasts for around 3 months, and about 10% of the hair are in telogen. So daily we will have around 10 to 100 hair falls per day from your scalp, and that is because of the catagen and the anagen phase. That is about 1% of the hair. The telogen hair is shed and the anagen hair replaces. Now the telogen, that is 10% of the hair, will fall every day, and it is obviously substituted by new hair. Coming to the sebaceous gland, the lipid producing, these are the lipid producing holocrine glands. It arises from the hair follicles at the junction of the infundibulum and the isthmus. It is distributed all over the body except the palms and soles. They are most numerous and large part in the face and scalp. Hence, the disease like acne and folliculitis we will see mainly in the face and scalp. We have a lot of hair and sebaceous, mainly the acne. We will have that in the face. They mature at puberty and are stimulated by various hormones. These sebaceous glands, they consist of lobules of epithelial cells that differentiate towards lipid-producing cells in a centripetal manner. Enlarged vacuolated cells in the center of the lobule disintegrate into an amorphous mass, which is called as the sebum. Now, this is a short question for you. If they ask what are the components of the sebum, that is the triglycerides, the wax esters, squalene, cholesterol esters, and the cholesterol. Now coming to eccrine sweat glands, these are mainly related with the thermoregulation. They are found in the entire surface of the body except the lips, external ear canal, and the labia minora. They are most concentrated in the palms and soles. And these eccrine sweat glands, they mainly open up in the hair follicles. We have the apocrine sweat glands, which are vestigial sexual organs. They are found in axilla and the anogenital region, and the mammary glands are considered as the modified and specialized type of apocrine glands. Now coming to the blood supply and the lymphatic supply of the skin, they have extended subdermal and dermal plexuses. I will show you in a picture. Now actually we have a bed, horizontal bed of arteries and venules in the dermis. We have two beds, and horizontal arteries and arteries goes up, and the veins come down from the horizontal, and these supply the dermis. We have the two horizontal beds in the dermis. Now there is a dermal plexus, there is horizontal, superficial, and deep plexuses connected by vertical communicating vessels. We have the cutaneous vasculature, which is important. The cutaneous vasculature is important for the thermoregulation, and the cutaneous lymph node parallels the blood supply. They are also found around the blood supply. What are the skin innervations by the nerves? We have, for light touch, we have Merkel cells of the epidermis, the Meissner's corpuscles in the dermal papillae. For the pressure, we have Pacinian corpuscles in deep dermis or the subcutaneous tissue. For pain, we have transmitted through the naked nerve endings located in the basal layer of the epidermis. For the temperature, we have the Cruz valves which detect cold, the Ruffini end organs which detect the heat. For heat and cold proprioception also located in the superficial dermis. Adjacent dermatomes often overlap, and it is important in the local anesthesia. So this is basically what you need to know about the anatomy of the skin and the appendages of the skin. I know I have gone very fast for the presentation because for most of you I had taught this in detail in the practical classes. I think on the last batch couldn't come and attend, but when they will come, we will teach you more in detail and we will show you in the board with diagrams everything about the anatomy. Basically, you should know this anatomy, and I think they have taught you in the first year also about the anatomy. So you know this, and I hope you get all the points that I have mentioned here. Thank you.